chapter 16 follow-up, that's Metabolic 1. And what we'll do is we're going to record our results. I'm going to ask you all to have one person per team come up and record your results, and that will help drive a class discussion. And then we'll describe the Chapter 17 Metabolic Test Part 2 that will be inoculated today. Excuse me. So you all will recall that we did the phenol red sugar fermentation tests. And the thioglycollate test for anaerobic growth. So what I'm going to do is just put the table up here, the table that we followed for your inoculations on Tuesday. And we'll just very quickly review how you're going to read those tests. And then we'll have you pull your tubes. When we say pull, you guys, that's just the jargon that we use, meaning that you go and you retrieve your cultures from the incubator. That's what we mean by pull. And do remember, you guys, you want to be careful not to shake your tubes, because those thioglycollate, you're going to be reading the pattern of growth. So you don't want to shake the cans or the tubes before you get them. So you recall last time, and this is just the table on page 142. We had our organisms, so we had Alcaligenes sputalis. We had Bacillus subtilis. And we had E. coli. And we inoculated each of these bacteria into 3 different PR sugar fermentation tubes. PR glucose, which you guys remember glucose is a monosaccharide. If organisms are going to ferment any sugar, it's going to be glucose. Okay, and then we have our disaccharide, PR sucrose. And we had mentioned how sucrose is a disaccharide made up of which 2 monosaccharides? Great lab practical question. Fructose and glucose. Good. So if an organism can ferment glucose, which additional enzyme must it make for it to be able to ferment sucrose? Excellent. Sucrase, dynamite. You guys are getting it. And then our last sugar was another disaccharide, lactose, milk sugar. And what are the 2 monosaccharides that make up lactose?
Glucose
and galactose. Good. And which additional enzyme must a microbe make if it's going to ferment lactose?
Beta-galactosidase.
Beta-galactosidase, right. So on the lab practice, you guys, if you just, like you're exhausted and all you can think of is bacterial lactase, you'll get half credit. But to get full credit, we want you to use the actual term beta-galactosidase. Good. And you guys recall that the PR sugar fermentation media, they have a little inverted glass tube. What's the inverted glass tube called?
Durham tube.
Durham tube, D-U-R-H-A-M. And what's its function?
Collect gas.
Collect gas, right? And what are 2 really common gases that are produced during fermentation?
H2 and CO2.
Good. H2 and CO2, so molecular hydrogen and carbon dioxide. Dynamite, you guys. Okay. In the phenol red, what is the... in the PR sugar fermentation media, what is the pH indicator? Phenol red, right, phenol red. And the whole concept here is that if your organism can ferment the sugar, you'll have acidic end products, the pH will drop, and what color will the phenol red turn?
Yellow.
Yellow, right? So yellow would be a positive test for sugar fermentation. And you guys remember we're going to score both the pH. So if the phenol red turns yellow, we know acid was produced. That's positive for fermentation. And then we also score for gas. So if there's no gas bubble in the Durham tube, we put a negative sign for gas. If there is a bubble in the Durham tube, we would put acid / what?
Positive gas.
Plus gas. Good. And gas production becomes important in helping to identify unknown microbes. Good. Now, if your microbe can't ferment the sugar, is it just going to starve? No. What's it going to use instead?
Amino acids.
The amino acids, good. And what happens when our microbes tear apart the amino acids? When they release the amino group, it gets released as
ammonia,
right? And ammonia can bind hydrogen ions and cause what the pH?
Increase.
Increases, right? What color does phenol red turn under alkaline pH?
Red.
Red, okay? So if your tubes are red, gently shake them, you guys, to make sure the cells grew. Because if you had a failure of transfer, if the cells didn't grow, you'd have to start all over again, right? You have to make sure your cells actually grew. If the media is red, we say it's alkaline. Okay, and there should never be gas. Right. Now, if you want to use conventional abbreviations, you guys, this is the shortcut. So acid, no gas, we put A, negative. Acid plus gas, we put A plus. What do we use for alkaline? K. Dynamite, you guys. So that's the shorthand for alkaline, and again, there'd be no gas. Okay. So you want to score, and what I'd like you guys is if each team could record their team results up here using a different colored chalk for your team, like you'd be blue chalk or orange chalk for your team, and if you record your results, that will let us drive a pretty brief class discussion. Okay. The last test you're going to test today is thioglycollate. And you'll recall that you used Alkalogenes faecalis and E. coli. Now, I'm going to tell you what the predicted results are, because thioglycolate is tricky, I think. Thioglycolate is tricky. And you guys will recall that thioglycolate reduces molecular oxygen to create an anaerobic environment. We said that there's an oxidation reduction indicator present. And so wherever oxygen is present, you'll have a green color. And you guys will recall that you inoculated the tube straight down and straight back up, and you were careful not to jiggle your loop because we want to keep this portion anaerobic. So this is a test to see if your organism can grow anaerobically or not. Now, one reason it's confusing is when you harvested your cells and you inoculated the tubes, your cells may still have been in log or exponential phase of growth. So you might have some cells down here growing, but we want good growth in the anaerobic zone. Okay, so we want a lot of cells down here growing before we're going to call it positive for anaerobic growth. Okay, now I think this can be a little bit tricky sometimes. So I just want you guys to know that Alkalogenes faecalis predicted is negative. So does not grow anaerobically. And we would predict our E. coli. We know E. coli can carry out aerobic respiration if oxygen is present. We know it can switch to anaerobic metabolism if oxygen is not present. It can switch to anaerobic respiration if nitrate is present, for example. Or if need be, it switches to mixed acid fermentation. So we're predicting that your E. coli should be positive for anaerobic growth. Okay, so that's our predicted results. Okay, just to hopefully help you out a little bit. Okay, so again, what we want you guys to do is, as a team, we want to get your safety gear on, because these are definitely opportunistic pathogens. We want to have our coats on, our shoes on. Want to pull your tubes. I would suggest you guys get your test tube racks out so you can actually see the tube. Share the results with your entire team. Record your results in your table and your lab manual, and then have one person come up and record your team results. And then we'll do a quick discussion of results, and then we'll launch into the new test you're going to inoculate today. Okay? Okay, so we'll let you guys get going.
I think it's